of objects into smaller groups that are to some degree similar to one another. This talk is going to look at the question, after clustering, how do you look at the results? And I'm going to mention some different types of clustering and clustering-like algorithms. And then inform you some different types of potential problems with clustering. And my other job is I'm a professor of mathematics. And so there I want to show you some things that are sort of odd about clustering. And then I'm going to give some examples of seeing in many dimensions. So K-means clustering is going to be my default clustering algorithm for demonstration purposes. And it has a weird dependence on initial conditions. And we found a way around that, which we call multi-clustering. Then I'm going to mention nearest neighbor technique, so we just have them on the table. Then I'll show you a clustering algorithm you probably haven't seen before. The guy that invented this clustering algorithm used it on trans-infinite sets to build error-correcting codes that were never realized or used for anything except to prove theorems. I once told him I found an application for his algorithm, and he just looked at me and said, "No, tell me you didn't." So, you know. And then I'll do some visualizations, and then we tripped over something really interesting. What it amounts to is, all of this arises from work I did maybe 15 years ago with a student that failed horribly. And that horrible failure keeps leading to new math, which is kind of nice. Anyway, K-means clustering is a technique for breaking collections of data into points to find my numerical values into K groups. You can also use it on non-numerical values if you have to do more programming. The algorithm is iterative. It makes several passes, refining the clustering of each pass, so it's not that fast. No problem if you run it on thousands of points. It's a little bit of a problem if you run it on millions of points unless you're somehow distributing it across lots of different processes. One big problem with it is the user has to choose K, and we found one way around that. There are several. I like our way, and I'll show it to you in a minute or two. And often you just try several different Ks. Give me 3 clusters, give me 4 clusters, give me 5 clusters, and see which one looks the best, or which one has the best in-cluster to between-cluster statistics if you're feeling like a statistician or whatever. The algorithm is also stochastic. The series of iterations are initialized randomly in a sense, and so running the algorithm again can yield different clusterings. Again, examples follow. And the behavior of K-means depends strongly on how you choose to measure the distance between points. I'm teaching first semester calculus again this semester for perhaps the 30th time. And one of the things that we keep having problems with is when I mention to the students that there are several ways to measure distance, they get all upset. But it actually matters how you measure distance. So, the basic algorithm, you have the number of clusters you want, a whole bunch of points, and it gives you a point and cluster assignment. So you pick some points to be the initial cluster centers, and over, you assign points to the closest center. The new centers are the mean point positions of the clusters, and you keep doing that until the points stop moving, or until it's been going on too long, because some bastard constructed an example where one point keeps jumping back and forth between 2 clusters forever. So you have to kind of start a timer. No, bad cluster. Now, you then report the clusters. So in this case, I took that data, which is contrived to be extremely friendly to K-means clustering, and the cluster assignments are given in colors here. So here's a funny thing about K-means clustering. You think, well, it just goes out in clusters, so it's going to return clusters, and those are like, these blobs. But the blobs have some constraints on them. In particular, a convex set is one where any line segment between 2 points in the set has to stay inside the set, and that tilted diamond has that property. But the horseshoe doesn't. You can find 2 points in the horseshoe so that the line joining them jumps outside of the set. And so it turns out that with K-means clustering, every single cluster is the intersection of a convex polytope with the data space. So if those blue things are the cluster centers, those cobblestones are all the points that would be assigned to that cluster center. And there's some other funny stuff here. Like, notice that, these cluster centers are really close together, and there are all these points that are almost equidistant from them and fairly far from them. So when you do K-means clustering, depending on how the data is distributed, you can actually get some fairly irregular results because of this, you must be the interior of a convex tile. And so that's an example where the math kind of jumps up and goes, whoop, screws things up. Okay, so the other thing is, K-means can go wrong. So there's my very K-means friendly data set, and I ran it a couple of hundred times, and 2 of the results took one of the spherical groups and split it in half. Now, would anybody like to guess why I put less correct rather than wrong as the label? Okay. Pardon?
You don't know the answer.
No, I know the answer. I built those sets. There's an obviously correct answer. Yes, clustering is exploratory. It proves nothing, except when the data is that contrived. Well, I'll get to that. Okay, so here's an example of data that's not really all that k-means friendly, the donut and donut hole data set. And when you do k-means clustering on this, it pretty much always discovers the central donut hole, but then it takes the donut and breaks it, since I asked for 6 clusters, into 5 pieces in a very arbitrary fashion. And it really depends on where were the initial cluster centers. That's what breaks up the donut. Now, notice that if 2 points are assigned to the same cluster, they probably belong together. And that's what I meant by less correct. Because I'd really like the whole donut to be one giant cluster. And it can't be, because there's no way it's one giant cluster unless it eats the donut hole at its center, because it has to be inside of a convex tile. Okay, you see that at least the problem I'm facing. So, there's a spacer. My chief data scientist, Wendy, keeps telling me I have to put in spacers, otherwise I talk too fast.
Thank you, Wendy.
Hey. Okay, so here's a different algorithm, which I call multi-clustering. So you pick a range of values for k, and this is the k and k-means. You initialize this big matrix over the entire data set, which, if it's a big data set, you store as a sparse matrix. Then over and over, you pick a k from your possible range of k's, you do k-means clustering, and for any pair of points that are put together in a cluster within a particular k-means clustering, you increment the strength of connection to the matrix, and you just keep doing it. Then you remove the weak edges, and connected components are clusters. So what I'm doing is I'm taking a whole bunch of k-means clusterings, and I'll explain in a minute why I want to have a range of k's, and I'm gluing them together. So these guys always form a cluster, so in the end they form a cluster, and there are relatively few edges crossing the annulus here between the donut hole and the donut. And with these guys, since those chunks kept overlapping one another, while this guy was never clustered with one of those guys, there's a continuous chain of connections around the donut, and I get the donut. The only problem is it's a little slow. Ah, yes, multi-clustering is slow. Okay, the range of values for k needs to have its low end larger than the number of clusters you want to detect, so it's sort of a lower bound. Now, if you want to find 4 clusters, then you probably take from 8 to 20 clusters or something. Having the multiple values of k reduces the chance of detecting idiosyncratic features of the data. Like, you see how those guys are kind of, somewhat separate from the others? If you always chose 15 clusters, it would be very likely that those guys would form their own cluster because they kept getting chosen. Whereas if you jump around, that spreads the connections around. If we normalize by the number of clusterings we did and force the connection strength into range 0 to 1, then it turns out the values you get are entirely stable. Doing many samples washes out the stochasticity. And a statistician showed me something he said was a proof, and I sort of believe him, that I'm approaching an actual asymptotic object here somewhere. So there really is a repeatable result if you do enough clusterings. And then the number of clusterings used and the range of k's are tunable parameters. So this is actually a family of algorithms, though they tend not to behave that differently from one another. So when we finish building the matrix, this connection strength matrix, it contains information about how strongly connected each pair of data points is. And you can use that in a number of ways. We can think of M, the matrix, as specifying a network on the data points with strength values for the connections. To get clusters, we pick a connection strength and delete all the connections that are not at least that strong. The number of clusters is the number of resulting connected networks. A cluster derived in this fashion has its own remaining network structure that can be used for analysis and visualization. And then finally, the cut plot, and don't worry if the definition doesn't make sense because there's a picture coming up. The cut plot is a graph of the number of clusters we get as a function of the connection strength we pick for deletion of connections. So, I took that 3-ring data set, and I went ahead and ran k-means multi-clustering, and I got my algorithm. And then if I start way over here at don't cut any edges, everything's connected. So there were some connections that jumped from the donut hole out to the donut, or well, between the two donuts, because this is a 3-ring data set. And then right here at a connection strength of about, we were together 20% of the time, it jumped up to 2. Then for a very long time, it stayed at 3 clusters, and then it jumped up to 4, and then to 5, and then it went off like a rocket, because what's happening now is individual points are starting to flake off. But the nice thing is, this gives you a very clear indication that there's strong support for there being 3 clusters, which is, something you don't usually get out of these algorithms. And here are the 2 and 4 cluster versions, and sure enough, that little New Zealand-shaped thing in the middle ring did flake off on its own. And when there were 2 clusters, it was the center 2 rings being glued together. So this actually sort of even makes sense. There's some other data sets that are not convex, have non-convex clusters, according to my human biased notion of what ought to be a cluster, and that this algorithm can handle. The clusters and k-means multi-clustering are the transitive closure of associations resulting from many applications of the basic k-means algorithm. While basic k-means produces convex clusters, multi-clustering washes out the restriction imposed by the distance measure it discovers connected shapes. If you use some other metric, some other distance, you'd still be getting convex groups. It's just the notion of convexity would be the notion of convexity for that other notion of distance, and those can look really weird. In addition, the basic data object places an edge-weighted graph structure on the data, which can be mined for additional information. This graph gives the cluster natural internal distances. Okay, the only problem with this is this is not yet big data, because it would take way too long to do this on a truly big data set. So clustering of any sort permits pattern discovery. It can be purely exploratory, what clusters did we find, or directed. So multi-clustering lets you find more complicated patterns than most other clustering algorithms. It yields structured clusters. If you have a collection of points that are interesting, you can ask multi-clustering for the clusters that contain those points for all cut weights. This is like drawing concentric circles or blobs of tighter and looser groups containing the points you care about. Since the clusters produced have an edge-weighted graph structure, you can use multi-clustering to produce networks containing points of interest, then hand off to other analysis tools. We've actually done that quite a bit. And multi-clustering performs data segmentation for you, if you want it to. Multi-clustering can also be used to perform a form of hierarchical clustering that has fewer stability problems. And I'm sort of unhappy that Mandy isn't here. I have a student who has been working on the problem of having clustering cough out stable results, and she's got some wonderful results, and maybe, in 6 months, she can give a talk here about this. Anyway, the multi-clustering has a lot fewer stability problems when you're doing hierarchical clustering. And that's an example of what I mean by hierarchical clustering. This was something we did, which was sort of odd. These are all various weird optimization problems. This is finding a statistical design. This is figuring out how to use a simple calculator correctly. The ones way up there at the top are standard numerical optimization problems. And the thing that was cool about this was we actually used a whole lot of different optimization algorithms on a whole lot of different problems and used those to generate associations. And what we got was a fairly sensible family tree. For instance, all the numerical things grouped together. That got the paper published. So I was happy because at the time I was only a professor. But this framework of making an association matrix is much more general. Any reasonable similarity metric can be used to generate associations. This means that the multi-clustering technique can be used to integrate dissimilar types of information, and the edge-weighted matrix encoded in the associated matrix is really easy to build these trees out of. You just take the 2 things that are most strongly associated, and they're the bottom level association. Then you associate the next 2 things that have the strongest connection that doesn't connect things that are already connected to one another. It's a simple algorithm. So, pause for questions.
What was the actual data set for those did I miss that?
There isn't one. So here's the thing. The ones at the top are, here's a strange numerical function. Please, as if you were a calculus student, find the high point. But the one that's labeled +1 recall store, what I have is this stupid calculator with a plus button, a store, a recall button, and the number 1. And the question is, what's the biggest number you can generate with 12 keystrokes? They have the property that every pair of points is in one triangle. And so these are wildly different optimization problems. So there really wasn't a data set. We weren't optimizing any particular thing. Rather, we were trying to build a family tree of optimization problems. And the reason for that is, those of you who saw Justin's thing on evolutionary algorithms 2 months ago in this forum, I actually studied the theory of evolutionary algorithms. And if I knew a family tree like this, I could give somebody good suggestions about which version of evolutionary computation would probably be the right one for the problem he's studying today. So that's an advisory.
So this wasn't generated by the multi-clustering?
That one actually was generated by version of multi-clustering, but I then took the association. Yes, it was. It would take a long time to explain that. Sorry. A different one. Anyone else? Okay. So, clustering speed.
Question.
Oh, sorry.
How did you go and organize those optimizations on that previous screen you did there?
How did I organize them?
Yeah, how did you go and get your algorithm to discover that?
I'm not sure I understand the question yet.
So you got these various optimization techniques, right? Is that what this is about?
Those are all different optimization problems.
Problems, okay.
And then I used 23 separate optimization algorithms. Would you like me to explain where I got 23 separate optimization algorithms?
If you can do it quickly.
What I did was I took one optimization algorithm, evolutionary computation, and then I placed a restriction on which creatures could mate with other creatures in the evolving population. That restriction was in the form of a geography. So what I did was I used 23 different geographies, some of them quite sparse, some of them quite dense. That was a cheap way of turning one optimization algorithm into a large number of different optimization algorithms, but we had as much as 4 orders of magnitude change in the time to solution for some problems by simply changing the topology in which gene flow was allowed within evolution. Did that roughly answer your question?
Okay.
And that's a whole different talk. So, k-means makes multiple passes through the data. It's not too fast. Neighbor joining, the thing I did to do this, once you have the data you're neighbor joining on, is actually quite fast. You have to compute a distance matrix, but it's just squared. And then multi-clustering is remarkably slow, though you can make it more efficient by playing games. And then I wanted to throw up these 2 definitions. If you're trying to classify data records, users, whatever, you can either cluster them and just look at what comes up, and that's unsupervised. You're just looking at what the natural associations in the data are. And then if something's supervised, you've actually got some examples, some things you know, and you're trying to learn to identify those things, like spammer. That would be something you would probably do supervised training on. Okay, so here's the simplest thing I know of that's useful for classification, k-nearest neighbor. What you do is you pick the number k, it's very different from the k and k-means, and some points and n dimensions, and some of the points have to have labels on them. And then what you do is for each point, you find the k-nearest examples with labels and take a majority vote. So the picture probably makes more sense. We know a lot of the points color already. We've got these other points that we, for some reason, don't know their color. But like the red guy here is next to 2 reds and 1 blue, so 2 beats 1, we think he's red. And like that one over there, all of his nearest neighbors are red. So this is a really simple algorithm, and it's really, really fast, assuming you already have some classified examples. So that's the sort of range of speeds. Now I want to go into something that's entirely new. So if you look at that diagram at the right, it displays 32 points spaced out very evenly in a square. It's possible to create such arrays of points of any reasonable size in almost any data space that can be specified to a computer. Square, rectangle, circle, surface of a sphere, etc. And in fact, since I made that slide, Matt has managed to get a paper published in which what we did was we took the customer's database. If it's large enough, you can use it as the data space. And that turns out to be hugely useful because if you don't know the geometry of the space, selecting points at random from the database will do. And very often it can be quite hard to sample uniformly from the space, even if you know its geometry. So what you do then is you take this thing, this is a point packing on the unit square of 32 points, and you just use those as if they're k-means centers and bin the data according to which point it's closest to. Since those things are well spaced out, it's actually not a bad clustering algorithm. And the other thing is, if it gives you bad results in one orientation, you can do something else. Now, that turns out to be all 8 ways you could pick up that group of 32 points and put them back down. And if you remember any group theory, that's simply the dihedral group of the square being applied to the square containing the point packing. But it turns out if you look at a hypercube, like let's say a 3-dimensional cube, you can swap around the axes, x for y, y for z, rotate x, y, z, or you can pass the faces of the cube through one another. So that means if you're working with n-dimensional data, there are n factorial times 2 to the n different forms of each point packing. Yeah, that goes fast, eh? And so you get a lot of these things. Now, the technique is very fast, but since the clusters are specified, in a sense, before seeing the data, they don't adapt to the data at all, the way k-means, for instance, does. And so it'll be a low-quality clustering unless we get lucky. We can do point cloud binning, ignoring small clusters, and then use the surviving point cloud members to initialize another technique like k-means, and that actually gives you a very much smaller number of possible outcomes for the k-means, and you get them much sooner. Then using a point cloud in multiple orientations to do many clusterings can be used as the basis of a multi-clustering. Instead of doing a lot of k-means clusterings, I do a lot of random hyperoctahedral symmetries of the point cloud and use those to build the associator matrix, which is less slow. I can't quite with a straight face say fast. And then the well-spaced point cloud could be selected from the data points. That's what I was mentioning. And like I said, we've gotten a paper published on this technique. Matt presented it in St. Louis, right? You know, and so that was good, except for the part where we were in Missouri. Okay, and pause. 4 questions. Not even what is that? Oh,
How about that? The whole section that you were talking about. Any ideas on linear chapters for non-convex spaces?
not off the top of my head, but that sounds like, you know, I'm a math professor, so yeah, cool question, thanks. So, wait a minute, if you have a non-convex space, then it may not be a good idea to do that.
Exactly. So in that sense, then if you want to apply that logic to your data points in a non-convex space, it's not going to give you that same result.
Sure it will. All you have to do is understand the geometry of the space and put it through a transformation that makes it convex. Which may be possible to do systematically, but let's not try and do it in real time. So, this is a technique that's just useful for processing high-dimensional data. So that's a picture where I took 5-dimensional data and slammed it down to 2 dimensions. And what it did was, so let me see, can I read those later? Yeah. The blue points correspond to human leukocyte type 2 antibodies and the red ones to type 1s. And the cool thing is though, while I didn't get a nice line separating them, if you just draw this curve, the technique I used to process the data completely separates these 2 types of biological sequence, which is sort of nice. And originally it was separating them in 5 dimensions, and then I was able to make a picture that separates them in 2. Okay, so here's a demonstration that might make more sense. I took all of the polyominoes made of 5 or fewer squares. So like this one is the cross and that one's the L shape. They're not quite Tetris pieces, though the Tetris pieces are in there. And they're like, there's the one with the unique one with 1, the unique one with 2, and then we've got the guy with 3 and the elbow with 3 and so on. But all of them with 5 or fewer squares are in there. I then measured the number of squares, the longest dimension, the shortest dimension, the number of corners, the number of concave corners, the inis, the perimeter, the longest line of squares anywhere in it, the number of dihedral symmetries, so the number of ways you can pick it up and put it down on top of itself, and then whether or not it had a 180-degree rotation. So that one has a 180-degree rotational symmetry, that one does, all those guys do. So I measured 10 quantities, and then I simply used the projection technique to make a 2-dimensional picture. So, is that a fairly reasonable grouping of those things that capture some of their properties? the linear ones ended up there. The really absurdly hypersymmetrical compact one ended off way by itself. And the ugly ass ones formed a group. And the L shapes ended up together. So I was kind of pleased with the way that came out. So how am I doing this? So it's not too difficult to do this. It's called nonlinear projection. If you did principal components analysis, you'd be doing linear projection. And if you do principal components on the polyomino result, you need like 3 views to get the same information you get with 1 view this way. So what the thing does, it assigns the points in a high-dimensional space to points in the plane. You got all these guys up in high dimensions, you give each one a point in the plane, and literally I start off generating those points uniformly at random in the unit square. And then I use evolution to improve them. So what I'm trying to do is to maximize the correlation coefficient between the distances in n-dimensional space and the distances in 2-dimensional space. And that's all, and it turns out evolution can do this fairly efficiently. There's some other efficient ways to do it, and they're built into the statistics package R. So if you look up non-dimensional multimetric scaling, is that the correct keyword for R? Or do they just have 50 names for it?
They call it classical multidimensional scaling.
Okay. And so the advantage of using an evolutionary algorithm is the flexibility to choose which distance you care about, or even change the quantity you're measuring in a transparent fashion. So for instance, if you only care about the correlation of the short distances, you get a very different picture from only caring about the correlation of the long distances, where if you care about all of them, you get a picture like the one I showed you. So between 50 and 1,000 points, it's really easy, in the sense that the evolutionary algorithm finds a good projection quickly. Below 50 points, the algorithm converges slowly, because the algorithm is very rough and has lots of local optima in it. And above 1,000 points, you need to play tricks just because it slows down a little. One thing that's weird, usually when you're running evolution, if you can afford a large population, it's a good idea. In this optimization, it's a bad idea. And it turns out that the primary mechanism of search is not evolution, but genetic drift. I can defend that statement if you're willing to sit through an hour and a half of biology with me. And this was something my chief data scientist actually discovered a while ago, this genetic drift phenomenon. And so it's common for the silly thing to locate the same projection over and over in several different orientations. That means differing by a rotation and a translation. But you can fix that if you just want to do a little more programming. And it takes a few minutes to sample out 30 projections of a moderately large data set. So there are 4 types of DNA that were created to all look random to most statistical tests, which contain some subtle patterns generated by large finite state machines. And this thing had no problem separating them into 4 categories. In fact, not only did I get the 4 categories, but the synthetic data I generated was from 150 to 250 DNA bases in length, so they got spread out like banners away from the center, which I wasn't expecting. So it's good for making diagrams of high-dimensional data when there are many relevant measured features. Changing the optimization target, like I said, only looking at the short distances, can give you different properties to the data. Nonlinear projection is not principal components analysis, rather it's a version of non-dimensional multimetric scaling. And so what's the difference? Suppose you had a data set where you had sort of, your data points were in a plane here, and then they turned a corner and went in the plane here, then turned a corner and went in the plane here, and this turn were in completely different directions because I can only show you 3 dimensions, but there's actually like 15 in your data. And this thing would flatten that, this would flatten it out into a big flag. If you did principal components analysis, you'd need at least 3 views to see those 3 separate planes. I'm not saying it's better, I'm saying it's different. And then we have another technique that I might like to come give a talk about in a year called agent case embeddings, but I've already given you one example of that. Take a bunch of optimization problems, take a bunch of optimization algorithms, and use those to build up the association matrix, and then build a family tree. So if you combine this nonlinear projection with that kind of tool, you can get some really wonderful tools for seeing relationships in your data. And you can also spike it. So there's a game called Prisoner's Dilemma. It's about how 2 guys that have been put in separate rooms by the sheriff and told he'll give them an easier sentence if they testify against one another. And we were studying a weird variant of that. How many of you have walked into a shop and said hello, and the person behind the counter goes, "What's it to you?" You know, that doesn't happen often, but it happens occasionally. Why? Can somebody give me a mechanism for that?
criticality? He's had enough of other people that day?
Okay, I'm not sure that's self-organized criticality, but the last customer was a jerk is precisely what we were studying. So what we did was we were training agents to play Prisoner's Dilemma, but instead of having them play one opponent for a long time, we kept changing which opponent they were playing. And we called that the shopkeeper variant. And then what happened is these different colors are different groups of customers that the general public is sampled from. Mix 3, Mix 2, Mix 1, Pavlov, Fortress 3, and so on. And then what we did was we spiked into the mix of our indicator variables the player that always is nasty, the player that is always nice, the player that does what you did last time, and his diametric opposite called Psycho, which is actually a very bad strategy, and as you can see, no points landed here. Okay. How we doing? Let's keep moving.
I have a question.
Oh, yes.
The DNA data, I'm not familiar how you get them. Are they a result of long reads or short reads?
That DNA data was synthetic. It was created by a finite state machine. I specifically created to create DNA that looked random in the sense that it had a Shannon entropy near 2, but still contained buried patterns. The HLA, the one biological data in there was taken from short reads that were then assembled on a scaffold. So they were probably really the immune sequences from a human being. Did that roughly answer your question?
Kind of, but then how you said you have different types, so you took from
different lengths.
You show different. You show 4 DNA samples, yeah. And,
so those are 4 different finite state machines, all trained to generate almost random DNA, but different finite state machines. So I was able to detect the residual non-randomness, and then it was just when I ran the finite state machine to cough out the data, I kept snipping off chunks of length 150 to 250 with the length chosen uniformly at random, which is what caused those bars.
So how this helps like a real-life application?
In that case, it helps a real-life application by letting me hand off to somebody a tool that I've certified has certain properties. That was the testing that we were doing to make sure it had the properties we needed. But like Andrew is currently using them to try and tell dehydrins from other disordered proteins, right? So what it amounts to is we have a technique of running DNA through a finite state machine that is evolved so that it will be in different states when different types of DNA are being fed through it. And this gives us a way of generating literally hundreds of thousands of different features on DNA. And we're currently using it to solve some fairly hard classification problems because we have these proteins that we think are involved in plants surviving drought stress. But there are 2 problems. We're not sure. And probably most of the proteins in that family haven't been identified yet because they're amorphous and have very little structure. So what we're doing is we're building something that can recognize the ones that we know exist. Then we're going to use it in the plant genome database to try and find more. And once we find more of them, one, we may start seeing patterns that will let us actually demonstrate they really do help with drought stress. And this is all part of Guelph's thing where they would prefer that as global warming or climate change happens that we all continue eating. So I like that application.
I have just one more question, a general one. I did some reading about this DNA stuff. I'm not a specialist, but like a general information and stuff. So it looks like there is no like 100% science in reading DNA, even the human ones. It's like kind of approximative. It's not settled. Then how you can use this method? I mean, you narrow down the problem, but it's not 100% sure. Yeah.
I call that biology. What you just described is true in almost all of biology. But so what you do is you use large numbers of samples, you sample across many individuals, you perform tests, then you re-perform the test to try and make sure they do what you think they do. This is why it takes 15 years to bring a drug to market, is because biology is very inexact and you can get sued for a lot of money, so you have to be cautious. But like my chief data scientist, what she did for her doctoral thesis was in part using these tools to locate all the DNA in the human genome that arose from viruses. And it turns out that 8 to 12% of our genome comes from viruses, and some of it's truly critical sequence. For instance, there's an immunodeficiency virus related to the AIDS virus that integrated into the human genome. and it's only expressed in the placental tissue of pregnant women, and it compromises their immune system. And if it didn't do that, the babies would all die at 5 months of allergic reactions to their mother. So extended human gestation is actually enabled by this weird integrated viral sequence. And we know it's a viral sequence because it looks a lot like other viruses, not because we actually found the virus. So all the information is sort of this, in DNA, is this reasoning by analogy guesswork. In fact, in some ways I kind of, my actual position at the University of Guelph is I hold a chair in bioinformatics, and my job is shoveling biologists out from under piles of data after they've dropped too much data on their heads through carelessness. And in a way it's kind of frustrating because before I got to just prove theorems and stuff, and now I have to deal with all this mess, right, Matt? I'm training Matt to do this too, and so it's kind of fun. And Rachel, though she's just starting out. Okay, that's a Julia set generated by 3 complex parameters. So if you know the right 6 numbers, they specify how to draw that rather complicated picture. Yeah, this time my spacer is actually something in the next data set. So I generated 1,000 different pictures that are interesting according to a measure of interest that I can tell you about if you care. And then I used nonlinear projection of where those groups of 6 numbers fell in essentially in a box that goes from -2 to 2, and 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 from -2 to 2, because all the interesting parameters have to be inside that box. And then this is the way that R will show you this data set if you just say, show me the data set. So what this is every 2-dimensional view, first dimension versus second, first versus third, first versus fourth. And this is the nonlinear projection from 6 dimensions down to 2. And I think it had a Pearson correlation of like 0.995, so that's a fairly accurate reproduction of the 6-dimensional distances in 2 dimensions. And as you can see, the information is just easier to see there, right? You can see where the clusters are. Okay, so that was sort of the end of that unit, so I need to talk faster. A graph is a very simple formalism for recording the connectivity of the network. There's an example of a graph. It has 10 vertices, it has 15 edges. It's called the pentagonal prism, if anybody cares, and it's a fairly logical name for it. So suppose we have a clustering of a data set, and we can measure the distance between the clusters somehow. Then we can define a graph G, in which the vertices of the graph contain the clusters, and the edges are created by placing an edge between a vertex and its 3 or other number of nearest neighbors. Vertices would be labeled with their cluster sizes. Adjacency in the graph means the clusters are near one another in the higher dimensional space, but the graph displays it as a flat picture. So this is a way to get a nice diagram of n-space data in 2 dimensions. Now, since the vertex may be one of the 3 nearest neighbors of a vertex that is not one of its 3 nearest neighbors, let's just say it this way, you are one of my 3 nearest neighbor relations is asymmetric. So some people will have more than 3 neighbors when we do this. And so this technique, like I said, makes a 2-dimensional picture no matter what the data dimension. Public domain code like dot inside of GraphViz can perform the graph layout. You can cluster several times to make multiple pictures of the same data, which gives you better perspective. And if you change the number of clusters, for example, it gives you structural information on the clusters. And then large clusters, like that 400 up there, you can simply pull them out and then redo the whole process to get a zoomed-in picture of what's going on there. So this is a nice way of doing hierarchical visualization. Now there is that data set. I'm trying to go slow enough that your doohickey can deal with it. Laid out by dot in GraphViz, showing you where all the clusters are with 36 clusters. And one of the problems is GraphViz tries to make a nice picture of the graph, but this guy is really close to that guy, but not in this picture. So that's a little sad. So instead, I'm going to use nonlinear projection. Now in this case, I've only got 8 clusters, so it's that data set, and now I know the relative distance between the clusters as well. And there is that same 8 cluster thing done with GraphViz's dot program and with this. Now, let's look. 149, 177, 161. 149, 177, 161. So GraphViz thought those made a nice triangle, and yeah, it's a triangle, but it's a triangle that's been hammered flat. in the real spatial relation. There I redid it with 12 vertices, and some funny things happen. Like if you look in the graph views, it's easy to see that the 144 is next to the 125 and the 118. But if you look here, first of all, the 425 got run over by the 47, which means I need to work on my layout algorithm. And 44 is that far from that and that far from that. So these real distances are very different from the distances we're getting out of the graph viz layout, though they both show you things that are true about the data. And almost done.
Sorry, how did you connect with an edge in that
Three nearest neighbors. So each guy is connected to the three other vertices closest to it. And for instance, with this one, his three closest guys were probably this one, and this one. But this guy was way out at the edge of everything. Where's 127? No, I'm wrong. So 37, never mind. Sorting that out takes too long. But yeah, it's just, you attach it to the three nearest neighbors, and if you want to, oh wait. No, except that this one was done with two nearest neighbors, because when I did three nearest neighbors, there were so many edges I couldn't see what was going on. So I can just ratchet that up and down, though if you go to one, it gets silly. Okay, that's the Mandelbrot set. It's probably one of the most famous fractals on Earth. And what I'm doing here is testing a strange evolutionary algorithm you might need someday if you're optimizing a model that instead of finding a good point in the model, finds lots of optima in the model. We found a representation called the contracting walking triangle representation, which is a cool name, but there's a time to explain it. They can actually enumerate optima and avoid parts of the space where it's already found an optima. So what I did was I coughed out the first 1,000 optima of I'd like to see a small Mandelbrot set inside the big one. And that's a simple thing to write a fitness function for, because what you get is a whole lot of points that take forever to diverge in the center of the view. And so they're the first 300 of the 1,000 optima I had the thing cough out. And as you can see, there are lots of little Mandelbrot sets in there. Here are the 24 best optima I found. And as you can see, they clearly have Mandelbrot sets. Now let's look at the middle 24. Well, yeah, there are Mandelbrot sets in most of those. And the last 24. And that means that given that I was only zooming between e to the -4th and e to the -10th for how much I enlarged the square, I probably found almost all the optima in that three-dimensional space, which is actually 3 by 3 by about 10 feet. And then, look at that. Look at that. This displays every single optima I found and uses colored crosses to tell you how zoomed in they are. Those are the sidelines from red to purple. When I do the nonlinear projection, it turns out, and this isn't intentional, that the long sidelines are up here and the short ones are down there. So you can sort of see the 3 by 3 by 10 rectangle in which all the optima were found. And I can even see now, like, well, there are not too many there, not too many there, there's a whole bunch there. That's a big pile. That turns out to be, never mind. I don't have time to explain an atlas of the Mandelbrot set, but it's a region where there ought to be a lot of these optima, and there were, so I'm happy. So, this is the one weird thing we found that we didn't expect. Does that tend to suggest that the number of optima have different density in different parts of the space? Well, yeah, there's big numbers. Remember, we're labeling the vertices with the number of optima that appeared in that group. So, yeah, there's a pretty big variation. So, in the graph-based display, we have labeled the vertices with the number of points presented in the cluster represented by each vertex. This gives us a sense of which parts of the space have high density of data and which have a low density. In the paper where we invented point packing techniques, without noticing we'd done so, we were point packing training examples to find a good lean training set for software agents. So what I did was I said, I'm clever, there are 250,000 training cases, I'm going to try and find 200. So if I train on those 200, I'll have a killer agent. And what I did was I found a well-spaced out set of training cases. And the agents were far worse than ones trained on 200 random examples. Why? Well, it turned out most of the normal training cases were all bunched together in one part of the space. So in the whole data space, I designed a killer bad training set. And that's what defeated us. And if I had had this point packing based graph display technique, I would have noticed much faster. we have a whole paper on figuring out why this didn't work. And it got accepted and everything. Normally when you have a negative result, but apparently if you render your garment spectacularly enough, they'll publish it. And then I'm a little sorry Jim didn't make it here tonight. There's a company I'm working with, Objective Motion. We're just getting started. And this is just another example of something we're analyzing with these techniques. This is the motion track of a robot that's picking up lots of little pieces of garbage. And all the robots are picking up the same garbage so that we can compare the robots. This is another example of this optimization algorithms, optimization problems. Robots, different fields of garbage. They'll let me understand the robots. Now, the very best robot we managed to train was this one. And when we do clustering, we find that we're actually finding something like that solution fairly often. And this one's slightly more optimized, and maybe a little harder on the robot because it's turning. This robot is the worst, and he's hysterical. First of all, he's facing the wrong way, and Jim actually did movies of these, and that one was sort of hysterical in a sad sort of way. And then this one, I mean, if my big concern were the robot not turning too fast, this would be my man, because he's like, it's very slow. And then this one, it has this herky-jerky, bang-bang control strategy. So instead of walking toward the garbage, it goes like this. No, here. Oh, yeah, what are you doing over there? No, that way. Again. But what we've got here is I've run 1,000 evolutionary trainings of these robots, so now I've got 1,000 different robot controllers that I'm using to understand how to cluster complex motion traces. So we can make clustering for the technical committee on games. And my chief data scientist is chair of the IEEE's technical committee on bioinformatics and bioengineering. So we both actually are, have fairly high status in the academic community in the sense that we do way too much volunteer work. But it means we know everybody. And then we're able to perform literature searches with far more than a keyword level of understanding. If you want us to do research for you, we'll come talk to you for a couple of hours and find out what you're really interested in. And we may be able to search better than your people because of all the people we know and all the things we know. And we can also design training courses if you want to learn about any of these topics we know about, or we'll research topics if we have to. We're happy to perform initial consultations to see if we can help you, and to help you focus your own understanding of your analytics needs if they are all unfocused, which hopefully they are. And our current staff includes 2 PhDs in math, 1 in computer science, and we work directly with dozens of students, and if it matters, we're veteran grant writers. I'm currently working on a couple of my tax grants to fund my students with Objective Motion. And if you need a university partner to do things, I'd like to be the Chief of Innovation of Ashlock and McGinnis Consulting. But if I need to be to land a contract with you or to work with you, I can be Professor Ashlock, in case that role is more helpful. Done. Wendy?
About 10%.
About what?
10%.
10%? Okay. I was watching a talk about that, and they were talking about how the virus makes up that percentage of the human genome, and they were saying that this was an accelerant of evolution for mammals and correlates to the mass extinction. And I just wanted to hear what your view.
I didn't know it was correlated with a mass extinction, but it might have accelerated that.
Yeah, and this is what I wanted to ask you was: is there any, is there any truth to that statement? As
But
they claim
she's the expert.
the rise of viruses corresponded to that time, and that didn't seem to be. I didn't.
Well, viruses are, ancient. They've been around and they continue to be around. It's not.
Well, this is what their point was, that the viruses are not seen in the record until that period of time, and to me that sounds false.
Well,
Yeah, that doesn't sound
6,000
right.
bases of DNA, what kind of fossil is that going to leave? 6,000 bases.
Yeah, well, you can see it in the genomes. the viruses that integrate it, and you can estimate how old they are.
And that
So
goes
now-
back into the billions, right?
Right, yeah, there's this argument, what was the first form of life? Was it a virus,
Virus,
or was it
yeah.
RNA? And so,
And if you intersect all the genes that every life form we know of has and keep simplifying, the ones that are the core that all life has are the genes that all viruses have, even though viruses are technically not really alive.
Right, okay. That's, I just didn't believe in that statement that the viruses did not come into the record until the
yeah,
mass extinction.
that's not right,
That
yeah.
makes seem false.
and humans are not unique in having viruses in their genome. All organisms
Yeah, no, they
have
didn't say that. It
viruses.
wasn't it was the acceleration of the whole, like the mammals after the extinction of the dinosaurs. Right?
But, for example,
yeah.
somehow the First Nations, with no real theory of genetics, turned something that looked like crappy wheat into corn in 11,000 years with a very simple breeding technique. That means it wasn't really the First Nations, it was the corn that was weird. And it turns out corn is 70% viral-derived genes. And those viruses pick up copies of working corn genes and make duplicates of them. And that's why you can do so much with corn. It has a mechanism for making lots of copies of its own genes, and then you can muck with some of the copies and not muck with the others. It's why corn is so selectable. And so that's an example of a huge viral genetic load that turns out to actually be useful. And yeah, it does accelerate evolution, except that now corn is a human commensal. We've so hijacked its evolution that if we died, it would die too. Can't reproduce without our help. Does anybody have any question on the less biological part?
So I'm interested in, I think you called it non-linear projections.
Yes.
And then the analysis, or the analysis when we talk about crystal component analysis, that's using computational time to reduce the number of attributes and number of features. Have you done any work where you use this projection to reduce or do feature selection?
So indirectly, yes, but we tend to use another method, which is we generate 100 million features, then we start taking subsets of the features and see which one of them work really well in a simple classifier.
Okay.
So we offload all the work into the feature generator, and then we just harvest good features, though since we take them in groups, they may be features that only work well together. Yes, you can use nonlinear projection for feature selection, and this would be much more useful for you guys than it would for me, because I'm always going, yeah, I guess I'll generate another million features and run the selector again. Whereas you guys have these exceedingly expensive features that you've already bought with blood. So yeah, that would be an interesting thing to work on if you wanted to put together a MyTech student and have a project or something. Anyone else?
Do you take another biological question?
Maybe my chief data scientist will, but go ahead.
Any progress of using machine learning to decipher the junk DNA?
Right
That
now,
is
we're fairly sure
90%?
there's no such thing as junk DNA. That is an idiotic designation made because we didn't understand how DNA works. It turns out, okay, so a long time ago, a mathematician proved that there is no multicellular life on Earth. And it was a correct mathematical proof, and the biologist said, well, he's obviously an idiot, instead of reading his paper, which they badly needed to do, because he found a real problem with biology. His proof goes like this. At the time, biologists thought that living organisms were run by their proteins. And if you have so many proteins, you need a traffic cop protein to keep them from interfering with one another. And the number of traffic cops you need goes up slightly faster than the number of proteins. And pretty soon the traffic cops need Mr. Inspector Supervisor traffic cops to keep the traffic cop proteins. And so he showed that if you have more than one cell, you can't have that many proteins. The control structure is top-heavy and it dies. Now, this was before we discovered small RNA. It turns out most of the control of proteins is not done by proteins. It's done by little bits of RNA that block the production of protein, encourage the production of protein, block the production of protein in special circumstances. And here's the thing. When proteins interact with proteins, it's an analog process. You have charge patterns on the surface of the protein interacting with one another in some way. If you have RNA, it's Watson-Crick base pairing, C to G, A to T. So it's digital. It's highly precise. And it's an enormously more useful control structure. And it turns out almost all the informational control of our cellular processes and the integration of cellular processes to make tissues and whole humans is done by these RNA molecules interfering with one another. And like, this has already got somebody a Nobel Prize, and it was a horrible shock, but it means there are, for every protein gene, there's dozens of RNA genes. And since RNA is really easy to break into pieces, one RNA gene may collide with another one and generate 15 control functions. But the only DNA we're sure is junk is genes that have gotten trashed by something like a virus-like particle inserting into them and making them unusable. But the fraction of DNA that's actually junk is way lower than we thought it was. Does that roughly answer your question? Oh, and machine learning is used routinely to spot these small RNA genes. So, in a sense, the answer is yes.
But, my notion of junk DNA, I was reading somewhere, it says that 90% of human DNA is junk. I mean, it's unknown.
But the truth is, about 2% of human DNA codes for protein. The rest of it codes for something else, and it was called junk under the hypothesis that protein was doing all the work. But that hypothesis turns out to be false. Wendy, did I get that roughly correct? Okay. Are we out of time?
Is it fair to say that is known? 90% is known? I mean, we know almost all?
No. We think we have an idea what's going on. To claim that we know what it's doing would be wildly overclaiming. We now know which rocks to look under. We don't know what's under the rocks.
Thank you. Last question. Last question.
So regarding clustering, so you mentioned very briefly that there is evaluation metrics to run on these clusters if you generate them without knowing decay or etc.
Yes.
So auto generation. Is there anything for this particular mechanism which improves on those trust measures in this, or the quality measures of the cluster? Or the regular methods are enough?
The regular methods are probably enough, but we don't need them because we have this proof that we're approaching an asymptotic truth. K-means is stochastic. To run K-means, you get different results. You need a statistical test to tell when you've got a good K-means. With our stuff, you run it long enough, and you're getting the truth ordained by God. This is what's in the data. There's nothing else in the data. Have a nice day. Now, you have to run for a long time before you get there. And at this point, I'm definitely math professor Dan, not data scientist Dan. Because, you know, anytime you use real data, something goes wrong.
I use this, I can trust my cluster.
So how long does that last?
90% yes.
If you run it long enough.
Okay, go.
And how do you determine that the clusters you generate using the data sets are relevant for anything useful? Because a lot of times you can get data sets that are
Clustering. So if the data set's contrived, it's not going to be useful for anything.
not that the data set's contrived, but like, for instance, I work in network security, and if you look at IP addresses, they're going to be clustered amongst things like local IP addresses.
Okay, so I can tell you what the clusters actually are. Meaning is your problem. Clustering is exploratory and suggestive. It gives you hypotheses. And in fact, this is something that infuriates my biological collaborators because they'll say, I found some cool clusters. And they'll say, what does it mean? And I say, well, that's your damn job. So, apologies, but I have a very unhelpful answer.
Okay, cool. Thank you very much. So thank you all for coming today. Drive safe back home. We're going to take a break for December. And
Oh, good. Driving here tonight was extra fun.
And then also a friend was mentioning that there's tomorrow a free screening of Fantastic Beasts, so if people want to get tickets for that, they can email me and I will message my friend. Thank you. And then the data science meetup is at the 27th of November. And is he still here now? The guy who was at the door earlier today, he's speaking here.
So I'm still trying to get